Hi everyone, this is Glenda Ganzon and welcome to my Human Anatomy and Physiology class. And for today's video, I'm going to be discussing the divisions of the autonomic nervous system. So stay tuned. The nervous system can be divided into two functional parts, the somatic nervous system and the autonomic nervous system. The major differences between the two systems are evident in the responses that each produces. The somatic nervous system causes contraction of the skeletal muscles, and the autonomic nervous system controls cardiac and smooth muscles, as well as glandular tissue. The somatic nervous system is associated with voluntary responses through though many can happen without conscious awareness, like breathing, and the autonomic nervous system is associated with involuntary responses such as those related to homeostasis. The autonomic nervous system regulates many of the internal organs through a balance of two aspects or divisions. In addition to the endocrine system, the autonomic nervous system is instrumental in homeostatic mechanism in the body. The two divisions of the autonomic nervous system are the sympathetic division and the parasympathetic division. The sympathetic system is associated with the fight or flight response, and the parasympathetic activity is referred to as the epithet of rest and digest. Homeostasis is the balance between the two systems. At each target effector, dual innervation determines activity. For example, the heart receives connection from both the sympathetic and the parasympathetic divisions. One causes heart rate to increase, whereas the other causes the heart rate to decrease. To respond to a threat, to fight or run away, the sympathetic system causes divergent effects as many different effector organs are activated together for a common purpose. More oxygen needs to be inhaled and delivered to skeletal muscle. The respiratory, cardiovascular, and musculoskeletal system are all activated together. Sweating keeps the excess heat that comes from the muscle contraction from causing the body to overheat. The digestive system shuts down so that blood is not absorbing nutrients when it should be delivering oxygen to the skeletal muscles. To coordinate all these responses, the connection in the sympathetic system diverge from the limited region of the central nervous system to a wide array of ganglia that project to the many effector organs simultaneously. So the complex set of structures that compose the output of the sympathetic system make it possible for these disparate effectors to come together in a coordinated systemic change. The sympathetic division of the autonomic nervous system influences the various organ systems of the body through connections emerging from the thoracic and upper lumbar spinal cord. It is referred to as the thoracolumbar system to reflect the anatomical basis. A central neuron in the lateral horn of any of these spinal regions projects to ganglia adjacent to the vertebral column through the ventral spinal roots. The majority of ganglia of the sympathetic system belong to a network of sympathetic chain ganglia that runs alongside the vertebral column. The ganglia appear as series of clusters of neurons linked by axonal bridges, and there are typically 23 ganglia in the chain on either side of the spinal column. So 3 corresponds to the cervical region, 12 are in the thoracic region, 4 are in the lumbar region, and 4 corresponds to the sacral region. The cervical and sacral levels are not connected to the spinal cord directly through the spinal roots, but through the ascending or descending connection through the bridges within the chain. The picture that shows the connection of the sympathetic system is somewhat like a circuit diagram that shows the electrical connections between different receptacles and devices. There are three different types of junctions that operate within the sympathetic system. The first type is the most direct. The sympathetic nerve projects to the chain ganglion at the same level as the target effector, which is the organ, tissue, or gland to be innervated. An example of this is the spinal nerve T1 that synapses with the T1 chain ganglion to innervate the trachea. The fibers of this branch are called the white rami communicantes. They are myelinated and therefore referred to as white, or the axon from the central neuron preganglionic fiber, shown as a solid line, synapses with the ganglionic neuron, with the post-ganglionic fiber shown as a dashed line. So this neuron then projects to the target effector, in this case the trachea, via gray rami communicantes, which are unmyelinated axons. In some cases, the target effector are located superior or inferior to the spinal segment at which the preganglionic fiber emerges. With respect to the wiring involved, the synapse with the ganglionic neuron occurs at chain ganglia superior or inferior to the location of the central neuron. An example of this is the spinal nerve T1 that innervates the eye. The spinal nerve tracks up through the chin until it reaches the superior cervical ganglion, where it synapses with the post-ganglionic neuron. The cervical ganglia are referred to as the paravertebral ganglia, given their location adjacent to the prevertebral ganglia in the sympathetic chain. Not all axons from the central neurons terminate in the chain ganglia. It branches from the ventral nerve root, continue through the chain, and on to the one of the collateral ganglia as the greater splanchnic nerve or lesser splanchnic nerve. For example, the greater splanchnic nerve at the level of T5 synapses with the collateral ganglion outside the chain before making the connection to the post-ganglionic nerve that innervate the stomach. Collateral ganglia, also called as prevertebral ganglia, are situated anterior to the vertebral column and receive inputs from splanchnic nerves as well as central sympathetic neurons. They are associated with controlling organs in the abdominal cavity and are also considered part of the enteric nervous system. The three collateral ganglia are the Celiac Ganglion, the superior mesenteric ganglion, and the inferior mesenteric ganglion. The word celiac is derived from the Latin word coelom, which refers to a body cavity, and in this case the abdominal cavity, and the word mesenteric refers to the digestive system. An axon from the central neuron that projects to a sympathetic ganglion is referred to as the preganglionic fiber or neuron and represents the output from the CNS to the ganglion. Because the sympathetic ganglia are adjacent to the vertebral column, preganglionic sympathetic fiber are related or relatively short, and they are myelinated. A post-ganglionic fiber, the axon from the ganglionic neuron that projects to the target effector, represents the output of a ganglion that directly influences the organ. Compared with the preganglionic fibers, post-ganglionic sympathetic fibers are long because of the relatively greater distance from the ganglion to the target effector, and these fibers are unmyelinated. Note that the term post-ganglionic neuron may be used to describe the projection from a ganglion to the target. The problem with the usage is that the cell body in the ganglion and only the fiber is post-ganglionic. Typically, the term neuron applies to the entire cell. One type of preganglionic sympathetic fiber does not terminate in a ganglion, and these are the axons from central sympathetic neurons that project to the adrenal medulla, the interior portion of the adrenal gland. These axons are still referred to as preganglionic fibers, but the target is not a ganglion. The adrenal medulla releases signaling molecules into the bloodstream rather than using axons to communicate with the target structures. The cell in the adrenal medulla that are contacted by the preganglionic fibers are called chromaffin cells. These cells are neurosecretory cells that develop from the neural crest along with the sympathetic ganglia, reinforcing the idea that the gland is functionally a sympathetic ganglion. The projection of the sympathetic division of the autonomic nervous system diverge widely, resulting in a broad influence of the system throughout the body. As a response to the threat, the sympathetic system would increase heart rate and breathing rate and cause blood flow to the skeletal muscle to increase and blood flow to the digestive system to decrease. And sweat gland secretion should also increase as part of the integrated response. And all of those physiological changes are going to be required to occur together to run away from the hunting lioness or the modern equivalent. This divergence is seen in the branching patterns of preganglionic sympathetic neurons. That is, a single preganglionic sympathetic neuron may have 10 to 20 targets. So an axon that leaves a central neuron of the lateral horn in the thoracolumbar spinal cord will pass through the white ramus communicans and enter the sympathetic chain, where it will branch toward a variety of targets. At the level of the spinal cord at which the preganglionic sympathetic fiber exceeds the spinal cord, a branch will synapse on a neuron in the adjacent chain ganglion. So some branches will extend up or down to a different level of the chain ganglia, and other branches will pass through the chain ganglia and project through one of the splanchnic nerves to a collateral ganglion. And finally, some branches may project through the splanchnic nerves to adrenal medulla, and all these branches mean that one preganglionic neuron can influence different regions of the sympathetic system very broadly by acting on wildly distributed organs. The parasympathetic division of the autonomic nervous system is named because its central neurons are located on either side of the thoracolumbar region of the spinal cord. The parasympathetic system can also be referred to as the craniosacral system or outflow, because the preganglionic neurons are located in nuclei of the brain or the brainstem and the lateral horn of the sacral spinal cord. The connections of circuits of the parasympathetic division are similar to the general layout of the sympathetic division, with a few specific differences. The preganglionic fibers from the cranial region travel in cranial nerves, whereas preganglionic fibers from the sacral region travel in the spinal nerves. So the targets of these fibers are terminal ganglia, which are located near or even within the target effector. And these ganglia are often referred to as the intramural ganglia when they are found within the walls of the target organ. So the post-ganglionic fiber projects from the terminal ganglia, which is a short distance to target effector or to the specific target tissue within the organ. Comparing the relative lengths of axon in the parasympathetic system, the preganglionic fibers are long and the post-ganglionic fibers are short because the ganglia are close to and sometimes within the target effectors. The cranial component of the parasympathetic system is based in a particular nuclei of the brainstem. In the midbrain, the Edinger-Westphal nucleus is part of the oculomotor complex, and axons from those neurons travel with the fibers in the oculomotor nerve or the cranial nerve 3 that innervate the extraocular muscles. So the preganglionic parasympathetic fibers within the cranial nerve 3 terminate in the ciliary ganglion, which is located in the posterior orbit. The post-ganglionic parasympathetic fibers then project to the smooth muscle of the iris to control the pupillary size. In the upper medulla, the salivatory nuclei contain neurons with axons that project through the facial and glossopharyngeal nerves to ganglia that control salivary glands. Tear production is influenced also by the parasympathetic fibers in the facial nerve, which activate the ganglion and ultimately the lacrimal or tear gland. So neurons in the dorsal nucleus of the vagus nerve and the nucleus ambiguus project through the vagus nerve, which is the cranial nerve 10, to the terminal ganglia of the thoracic and the abdominal cavities. So parasympathetic preganglion fibers primarily influence the heart, the bronchi and esophagus in the thoracic cavity, and the stomach, liver, pancreas, gallbladder, and small intestine in the abdominal cavity. So the postganglionic fibers form the ganglia activated by the vagus nerve are often incorporated into the structure of the organs such as the mesenteric plexus of the digestive tract organs and the intramural ganglia. So this ends my discussion about the division of the autonomic nervous system. If you have any question, please write it in the comment section down below and I'll be glad to answer them all. And if you're still not subscribed to my channel, please also do consider subscribing and hit the notification bell so that you'll be updated with my new videos. And if you like this video, please give it a thumbs up and share. Once again, I would like to thank you all for watching my video and for listening to my discussion. So until my next video, bye everyone!